So when we are focusing on the process of replication, here what we are learning, what we are going to learn, is how the DNA itself is duplicated. So this means that the entire genome is completely and faithfully duplicated uh, at every stage of the cell cycle. So at this point, I would like you to appreciate the enormity of this task that is constantly carried out. So we have enough letters of DNA, the A, T, G, and C in the entire genome that can fill up a million pages of a book, right? So it's an enormous length. And this length of DNA, the entire length of DNA, needs to be duplicated faithfully, without mistake. When I say faithfully, I mean without mistake, every time a cell divides. With transcription, it's easier, in my mind at least, because only a very small part of the genome is transcribed, right? Only about 1%. But when we come to replication, replication involves the entire genome, even the heterochromatin parts, the euchromatin parts, of course, but the heterochromatin parts, those parts that are not expressed at all, also have to be replicated. So this is an enormous task, and it entails a very, very high amount of activity of the DNA polymerase. So we're going to start with an overview of the process of replication. And in these videos, the primary focus is on the prokaryote, but we will also talk about the eukaryote at the end of the lecture. So let's get started. So in this, we first have an overview. Yes, conceptually it's simple because there isn't as much regulation involved in DNA replication as there is in transcription. We have both strands need to be replicated. Okay, so that's one important point. In transcription, remember, we had a coding strand and we had a non-coding strand. We had a template strand or a non-template strand. Whereas in replication, both strands need to be replicated. Now, one important point to keep in mind here is that the strands are complementary to each other. So at this point, your knowledge of what is the 5' end and what is the 3' end of DNA is very, very essential. So the 5' end of the nucleotide has the phosphate group. Remember, this is the 5th carbon of the ribose sugar, and the 3rd carbon of that ribose sugar has the hydroxyl group. So the 5' phosphate and the 3' OH refer to the phosphate group and the hydroxyl groups that are found in the nucleotide. So because each of the two strands are complementary to each other, each strand can serve as a template for the reproduction of the other. So both of them are templates. If we know the sequence of one strand, we can determine the sequence of the other strand because they're complementary to each other. So here is how the DNA replication is working. So we have the template here, and this is the new strand that is being formed. So in transcription, remember I was just mentioning, in transcription, where we have the non-template strand and the template strand, this is showing transcription actually, we have the separation of the two strands, and then one of the strands will act as the template for the synthesis of the RNA. So this can be ribosomal RNA, messenger RNA, doesn't matter. So only one strand of the DNA acts as the template. Whereas for replication, both strands are used. And both strands are replicated at the same time. So this sort of replication is called semi-conservative because when we look at the new cell that will be formed, now you're already aware of the cell cycle. So you already know that after the S phase, we will have the G2 phase, and then we will go to the M phase. And at the end of the M phase, we will have the cytokinesis giving us the two new daughter cells. So each of these two daughter cells will receive one strand of DNA from the parental cell and one strand of DNA, which is the newly synthesized strand. Okay, so that's why this is called semi-conservative, because half of the original DNA is kept while the other half is newly synthesized. So the original template strand is referred to as the parental strand, and the newly synthesized strand is referred to as the daughter strand. So this is called the daughter strand. Now there are a couple of points to keep in mind here, which is absolutely essential for you to never in your life make a mistake. The new strand, the daughter strand that is synthesized in a 5 to 3 primed direction. Logically, this is very, very easy. This is not something that you should be memorizing. You should be thinking of it in terms of logic. Because when you add a new nucleotide for the formation of the phosphodiester bond, that phosphodiester bond is formed between the 3' OH of an existing DNA strand and the new nucleotide, it's 5' phosphate of the new nucleotide, right? So between the 5' phosphate of the new nucleotide and the 3' OH of the existing strand, there will be a phosphodiester bond. So therefore, we always need a free 3' OH in order to add a new nucleotide. And therefore, the synthesis of the new strand will always have to be in a 5' to 3' direction. So that's one important point to keep in mind. So always the new strand has to be in the 5 to 3' direction. But this gives a conundrum, which we will address later on, because the two strands of the DNA are anti-parallel to each other, which means that one strand, if I were to label this strand, this upper strand will be 5 to 3', this lower strand will be 5 to 3' in the opposite orientation. So because the two strands are anti-parallel to each other, DNA replication will occur in a bidirectional manner in a very, very unique and special way. So this is the parental strand, this is the daughter strand. So the synthesis of the DNA is 5 to 3' of the new or the daughter strand. Okay, not of the parental strand, of the daughter strand, because the new nucleotide is added to the 3' OH of the existing DNA chain. Right. So now we come to the actual process of replication. So we are going to be looking at these videos, and then later on I'm going to go back to explain to you some of the proteins that are involved. So this is the one that comes after this. Let's have a look. So in the initiation of DNA replication is much easier to understand in prokaryotes compared to eukaryotes. So eukaryotic initiation of replication is a little bit more complex, simply based on the fact that there is a huge amount of DNA in eukaryotes. You appreciate that, of course. So this is a nucleus of the eukaryotic cell, and you can see that the enormous amount of genetic material that we have here. And remember, each and every nucleotide of this genetic material needs to be replicated in order to faithfully copy this cell into the two daughter cells. The prokaryote, on the other hand, has a single circular DNA, which is also several hundred nucleotides long, but that's about it. It only has a single circular DNA. And so it's far, far more simple to understand the initiation and continuation of replication in the prokaryotes. So in this particular video, we are going to focus solely on the prokaryotes, and then we will look later at eukaryotes as well. So the first step in replication is the initiation phase. Just as we learned with all other processes, the first step is the initiation process, and that process is critical because it will define what direction the process will proceed. So in the case of prokaryotes, in the case of bacteria, for example, the initiation of replication happens at a structure called ori, or origin of replication. Okay, so this is a specific DNA sequence called origin of replication, and to that origin of replication, an initiation protein will come and bind, and it will basically provide information to the DNA polymerase that the origin of replication is over here, and the DNA polymerase can now come and it will then start synthesizing both the strands of this DNA. Now, remember, prokaryotes have a single circular DNA, so each circular DNA or each bacteria therefore has only one origin of replication. Okay, each bacterial chromosome, there's only one chromosome. It has only one single origin of replication. Eukaryotes have multiple origins of replication on each chromosome, and we will look at that later. Okay, so this is the origin of replication. And so eukaryotes have many, prokaryotes have one. And the binding of this of the proteins that are going to show to the polymerase that this is where the origin of replication is, it will distort the shape of the DNA. Okay, so it distorts the three-dimensional shape of the DNA in such a way that the two strands, which remember have many hydrogen bonds, so they are all very closely intertwined with each other in the form of the double helix, but those numerous hydrogen bonds now need to be cleaved, right? The hydrogen bonds need to be broken. So the initiation proteins are basically helicases. Helicase, an example of a helicase is DNA A in bacteria. So this helicase will then, with the help of ATP, this is an energy intensive process, is going to separate the two strands, denature the two strands by breaking the hydrogen bonds. This results in the separation of the two strands and the formation of a replication fork. So what you are seeing here are many different proteins that we are also going to be looking at later on. But just to give you an idea, these are the single strands of DNA that have been pulled apart, that have been denatured with the help of the helicase function of at the origin of replication. Now, remember, these two strands can actually go back and recombine, right? This is what is happening during renaturation. So in the laboratory, you could do this very easily by heating up the DNA to a temperature of about 94 degrees for about 5 minutes, and then you allow it to cool down gradually to room temperature. So when you heat up the DNA, that heat will cause a breakage of the hydrogen bonds, and then when you cool down the DNA, the two strands of DNA will find each other, the complementary strands, of course, will find each other and they will reform the double helix. So in a living cell, obviously we can't heat it up. So what we do is we use helicase, and the ATP that is broken down by the helicase provides the energy to pull the two strands apart. And to keep these two strands apart, these are called single strand DNA binding proteins, or SSBs, that will go and bind to the single strands and keep them apart until they have been replicated. So this is called a replication bubble because it really looks like a bubble. And then we have the recruitment of, so here we have the bubble, and then we have the recruitment of the proteins, which will then lead to the replication. So we have the single strand of DNA here. This is called the fork, replication fork, and these are the nitrogenous bases, which will then be replicated with the help of with the help of the polymerase. So the opening of the replication bubble, it exposes the nitrogenous bases. Nitrogenous bases, remember, ATGC, these are the bases, along the inside edge of the nucleotides in that region. So they are all facing towards the bubble. This allows the recruitment of two large protein complexes called replisomes. So they attach to two ends of these bubbles and they move in the opposite direction. Okay, this makes it very efficient and very fast. So one thing to keep in mind, remember, is that the whole genome of E. coli, for example, needs to be is replicated within 40 minutes. So this is very rapid, very fast. So these two large complexes called the replisome, which has the DNA polymerase and several other proteins that we will learn, they bind to the replication bubble in the opposite orientation and then they move in opposite direction. So here we have, this is the replisome now. So within the replisome, we have a protein called the DNA polymerase. So it has two DNA polymerases, one for each of the two strands, right? So we have, remember, the two parental strands, and each of these two parental strands are going to be replicated by two different polymerases, two copies of the polymerase. Sometimes they can be different polymerases as well in eukaryotes. In bacteria, it's the same polymerase, but two copies of it, okay? One for each of the two strands at that fork. And there are several accessory proteins that we are going to focus on later. They're not shown or described very well in this in these videos. But maybe I should take this opportunity to tell you, to describe to you what these proteins are. So here you're seeing this circular protein. This is a protein called a beta clamp. So what the beta clamp does is you can see that it has a hole inside, and the diameter of this hole is about 2 nanometers, which is exactly the diameter of a single strand of DNA. So it basically, this beta clamp is like a ring. You know, if you can imagine a wedding ring that a person wears, it fits very nicely into the finger, but it can also be opened if necessary. So this beta clamp will clamp around the DNA, but it is also free to move along the DNA. And what this beta clamp does is it's capable of binding to a vast number of proteins. So it's also called a tool belt. So if you can imagine, you know, those workers who come for, you know, construction workers or plumber who comes to your house to repair things, they tend to have a belt which has many different tools in them, right? I'm sure you've seen that. So it's called a tool belt. So the beta clamp is likened to a tool belt whereby it has the ability of protein-protein interactions with many different proteins, which will then be recruited in a very, very organized manner to the DNA in order to carry out their function. So the beta clamp has a really important role in that. So here you go. It is coming. So now we have the polymerase being recruited, and the polymerase, as you can see, is moving with the beta clamp along the DNA in the replication bubble. And you can see the new strand here, which is shown in light blue, that has been synthesized. And this whole complex is called the replisome. To keep the chain separate, there is a helicase in protein synthesis. For protein synthesis, you're talking about mRNA being translated into protein. So there is no helicase there because there are no two strands. mRNA is a single strand.
Hocam, we separate the DNA chain also in protein synthesis, right? And
The replisome. Okay. So once the initiation has been carried out, which means that once the origin of replication has been identified, which, as I mentioned, is a specific sequence in the chromosome of the bacterium, the replication is ready to begin. The two replisomes, these are complexes of proteins consisting of DNA polymerases, they start moving away from each other in the opposite direction as they replicate the DNA. So this is almost like opening a zipper. So like the zipper we have in our jacket, it's almost like that. So we will see some pictures now, very nice pictures. So the replisomes will move in the opposite direction. And here you can actually see, it's a little bit difficult to understand from this video, but you will see how the two strands, so this is the new strand, this is another new strand, and this on the left is the parental strand. So you should be going from the left to the right. The parental strand is being separated by the helicase. And again, I'll repeat myself, helicases are found very, very frequently, and they are responsible for moving two strands apart, whether they are self-complementary regions in RNA or whether they are the DNA helices, the two strands of the DNA. In each case, a helicase is used. So the helicase has separated the two strands. And then in the replication bubble, we have the replisomes. And the replisome is, it has the DNA polymerase, which is replicating the DNA in both the strands. So remember, I mentioned, and we'll talk about it in the next video, I mentioned that the parental strands are in the opposite orientation. So they reverse complement of each other. So one is going in the 5 to 3 prime direction, the other is in the 3 to 5 prime direction. But the new strand can only be synthesized in the 5 to 3 prime direction. So therefore, one of the strands will be synthesized continuously. The other strand will be synthesized in bits and pieces. And that's something that we will focus on in the next slide. But here you can see the replisome, you can see the single strand DNA binding proteins, which is going to keep the single strand apart. It will prevent re-annealing. So this is like a zipper that is opening. The replisome in the zipper is the zipper handle that moves, and the double-stranded DNA is the closed part of the zipper, and the single-stranded DNAs are the new strands that are being synthesized. So the fork is not a fixed structure, obviously. The fork is something that is moving along the DNA. It's a transition point that moves along the replisome. And just like a zipper, each replisome will separate the double strand because of the presence of the helicase, remember, in that replisome. It will separate the two strands, and it will keep and then the single-stranded binding proteins will keep the single strands apart until they have been replicated. Now, one important point here, I'll take this moment to make you perhaps think about if we are considering not a circular chromosome, but a linear chromosome. And we have this sort of replication fork that is moving along the DNA. Would you expect the DNA on the left of this picture, so somewhere here, to undergo supercoiling, positive supercoiling? So again, I would like you to imagine a rubber band that you are separating with your fingers into the replication bubble, and you're holding one end of the rubber band. Okay, I don't have anything here to demonstrate it for you. Sometimes I have rubber bands, right now I don't, but you will have to imagine this. You're holding one end of the rubber band and you've twisted it, similar to how a double helix looks. And then you open up the other end of the rubber band with your fingers to mimic the replication bubble. So the part that is above the replication bubble, which is represented in this part, is expected to undergo supercoiling, right? So this supercoiling it needs to be relieved, and this is primarily seen in eukaryotes. It's not seen in prokaryotes. So there is a mechanism of supercoiling indeed during replication. And in this complex, therefore, we also need to have a protein, such as a topoisomerase, which can cut either a single strand or both strands of DNA, depending on the type of topoisomerase, which will relieve this supercoiling. Okay, so that's something I wanted you to appreciate, that this is not as straightforward as a zipper, because remember, there is a coiling of the DNA. So this is a very, very simplistic picture. So over here, we expect to have positive supercoiling, which needs to be relieved with the help of the topoisomerases. So the molecular machine at the front of the replisome that separates the double-stranded DNA into two single strands, as we mentioned, is a helicase. Each of the two DNA polymerase enzymes then uses one of those strands as a template upon which to synthesize a complementary strand of DNA. So this is the helicase, and it is opening up the two strands, and we have the polymerase here, we have the polymerase here. And at the background, I don't know if you could see the SSBs as well. They were not very, very clear. The DNA polymerase is extremely accurate. I'm going to talk about this a little bit later as well. And it rarely makes mistakes. And I'll explain to you how it prevents from itself from making mistakes. It makes less than one mistake in a billion. So when a mistake does occur, the DNA polymerase has the ability to proofread and fix that mistake. But I'm going to explain this in more detail later on. Let's look at the videos first. So now we come to the issue of anti-parallel nature of the parental strand. So we are going to learn how, so I'm going to stop sharing for a moment, and I will share the whiteboard with you just to make you appreciate what is the problem that I am trying to describe here. So if I draw, and I would encourage you to do the same, you draw a strand of DNA, right? And you label it as 5' to 3' and 5' to 3'. This is your parental strand of DNA. Now, what I mentioned is we will synthesize DNA in a 5' to 3' direction of the new strand. So maybe I could right? So this is the correct labeling. So when we look at the strand, I mean, this strand also should be in purple color. So you can imagine that. So when we look at the new strand, so this is the new strand, this is the new strand, this is the old strand, and this is the old strand, parental strand and daughter strand. So when we look at this particular strand, the synthesis can happen continuously in a 5' to 3' direction, no problem. But when we look at this other strand, when we look at the upper strand here, then the synthesis will occur in a step-by-step manner because it needs to go back upon itself and then it will need to synthesize it. So it cannot synthesize in a continuous manner this way. Rather, it needs to go back a little bit, go back about 150 nucleotides in the case of eukaryotes, synthesize a strand and come back and synthesize the new strand again. So we will see that problem being addressed in the next video. The anti-parallel. So each of the two parental strands acts as a template for the polymerization of the new daughter strand. And DNA polymerase can only synthesize in a 5' to 3' direction, which is same as for RNA polymerase, right? So they can only add a nucleotide with a phosphodiester bond to an existing free 3' OH. So if this is the parent strand, then the new daughter strand that is being synthesized, let me go back a step here. Oh, if I go back, it freezes. So here we go. Maybe this will help us. So the parental strand is over here, and this is the parental strand. So the new daughter strand, when it is being synthesized in a 5' to 3' direction, it's okay. It can proceed and it can be continuously synthesized. But remember, the polymerases are in the replisome. They are in the same location. They are not moving away from each other. And the polymerases were shown only to add nucleotides to a free 3' OH. They cannot add nucleotides anywhere else, new nucleotides, okay? So this daughter strand is going to be synthesized in the opposite direction. So how come this particular polymerase, which is located here, how will it synthesize in the opposite direction? But it has to synthesize in this direction because that's the only activity that it is capable of carrying out. So the chemical energy that is required to join the incoming nucleotide to the growing chain is provided by the nucleotide itself. So remember, we have the ATP or the GTP or the the CTP, whatever nucleotide is coming, it has the phosphate. So one of the phosphates, the gamma phosphate, is released. Actually, the two phosphates, the PPI, is released, and the beta and the gamma phosphates are released. And those two phosphates, when they break down, they provide an enormous amount of energy, which enables this reaction to proceed in a forward direction and not in the reverse direction, right? So this energy is provided by the nucleotide itself. So because of this constraint, the DNA polymerase must read the template strand in the opposite direction. So this presents a very big problem, right? Because how will the same polymerase carry out the function at the same time? So when the daughter strand is being synthesized in a 5' to 3' direction, it can be done continuously. This is being read in the correct direction. But then how about this polymerase? So how can we make sure that these two polymerases, even though they are stuck with the same replisome, how can they carry out this function at the same time? They are not moving away from each other. They are incapable of moving away from each other. So this means that there has to be something else going on that will enable them to replicate at the same time. And we will learn that here.
DNA replication is semi-discontinuous.
So here we go. So one of the strands is being replicated continuously in the 5 to 3' direction, and the other strand is being replicated semi-discontinuously. So when one DNA polymerase travels towards the replication fork, okay, so this is the DNA polymerase that is traveling with the replication fork. It travels down the correctly oriented strand, referred to as the leading strand. So in this case, the new strand is being synthesized in the 5 to 3' direction. The parental strand is in the 3 to 5' direction. The other polymerase travels away from the fork as it travels the correct way down in the other strand, in the opposite strand. This is referred to as the lagging strand. So here we are. It jumps back, synthesizes this part. Yeah. So when the camera is moving with the fork, this is what happens. So because the lagging strand polymerase moves backwards with respect to the replisome and replication fork, it cannot keep up with the newly emerging lagging strand DNA, that is the leading strand DNA that is being continuously generated here. So this is continuously generated. The other one, let's see, this is a very long drawn picture. It could be a little bit more. Okay, so this is the helicase. So the lagging strand polymerase is therefore forced to polymerize one short fragment at a time because it keeps having to go back to the start of the next fragment every time it completes the current fragment. So this one will therefore move back as to keep pace with the helicase, which is opening up this replication fork. So it will synthesize in this direction, which is the 5 to 3' direction here, and then it will go back and synthesize again, and it will go back and synthesize again. There you go. So therefore, unlike the leading strand polymerase, which never has to dissociate from the DNA, the lagging strand polymerase is forced to repeatedly associate and dissociate from the DNA polymerase as the daughter strand is synthesized. So for each, Gizem is asking how many base pairs does it complete for each fragment? This has been counted as about 100 to 150 for eukaryotes. It's a little bit longer for prokaryotes because prokaryotes need to replicate within a very short time. So the lagging strand polymerase actually comes back for many more nucleotides in prokaryotes. It's more efficient compared to eukaryotes. But in eukaryotes, it has been identified as about 150 nucleotides. So these fragments, as you know, are called Okazaki fragments after the biologist who discovered them. And they are a few thousand nucleotides long in prokaryotes. So because it's occurring continuously on the leading strand but discontinuously on the lagging strand, that's why DNA replication is said to be semi-discontinuous. In reality, the lagging strand polymerase doesn't really move away. It's a part of the replisome. So there you go. Now it will be easy to understand. So the DNA itself loops around. This is fantastic. So as the replisome moves forward, the loop grows larger from both ends, right? So this loop is growing larger. Fed at one end by the single-stranded DNA that is exiting the helicase, so that's this end. And then at the other end, the polymerase is going to be replicating it. So when the polymerase reaches one end of the Okazaki fragment, it releases the DNA and binds to the next part of the template so that the next Okazaki fragment can be generated again. This cycle repeats down the entire length of the DNA. And when we add all the specific proteins, then this is how it looks. So the leading strand is over here and the lagging strand is over here. So the upper polymerase synthesizes the new strand slower no, it actually isn't slower. Because if it was slower, then it would lead to a structural constraint that would be very difficult to cope with. So rather, the jumping motion actually, it does not cause any lag. It's called a lagging strand indeed. When you hear the word lagging, you would imagine that there is a delay, but there isn't any delay as such. It's just that it's happening in this discontinuous manner. But overall, the two strands are replicated more or less at the same duration, at the same speed. Okay, so we will look at this and then we will go to the some text. So this is the replication fork. The two forks are moving in opposite directions. Remember, we have two replisomes which are moving in the opposite direction. And this is how it looks. This is in prokaryote, obviously. This has a single circular DNA. In eukaryotes, there are many replication bubbles. So each chromosome can have hundreds, sometimes thousands of replication bubbles because it's so much bigger. So each replication bubble also moves in the opposite direction. So again, you have a leading strand and then you have a lagging strand. Both are seen here as well. And so let's look at these videos. So as they move, they merge with each other, and then we will have the complete DNA replicated. So in eukaryotes, each duplicated chromosome remains physically attached at the centromere. So remember those proteins we learned about last week. They are called cohesins. They keep the two strands together, and then when they're cleaved at anaphase, they will be separated. Okay. So now what I'm going to do is, for the remaining 10 minutes of the class, I am going to go back to our slides, and I'm going to talk a little bit more about what exactly is needed for replication to happen. So you have a basic idea of how replication occurs, but now what we need to understand is some of the more nitty-gritty details. So you've already learned that the replication happens in a 5 to 3' direction. So what this means is, for the polymerase to be able to add a new nucleotide, it needs a free 3' OH. The other thing that it needs, the second requirement that it has, is it needs a template. Okay? So this template is necessary for the polymerase to understand which nucleotide to add, right? So the template will provide information of which nucleotide to add, and the complementary nucleotide will be brought in. So A will bind to T, G will bind to C. So we need the 3' free 3' OH, we need the template. So therefore, this particular DNA will not be replicated unless it is separated with the helicase. But this one has the 3' OH and it has the template as well. This one also has the template, right? We have the 3' OH and we have the template. This one doesn't. This particular DNA strand does not have the free 3' OH. It has the template, but it does not have the free 3' OH, which brings me to a very important protein called primase. DNA replication can only happen, the polymerase can only catalyze the formation of a phosphodiester bond to an existing 3' OH. So in the case of DNA replication, we have to have a primer that will be attached here, and this is usually an RNA primer, which will give us the 3' OH, a free 3' OH, which will then be used by the polymerase to bring a new nucleotide, and the 5' phosphate of that new nucleotide will then form a phosphodiester bond with this free 3' OH with the release of water and with the release of the pyrophosphate PPI, and then we will have an addition of one nucleotide to this. So we will need a primer, and this primer, as I mentioned, is usually an RNA primer, which is laid down with the help of a protein called primase. So here we have a primer, which is giving us the 3' OH, and then we have the nucleotide coming, and this is the 5' phosphate. So we have the phosphodiester bond formed here. The two pyrophosphates, the two phosphates here, the beta and the gamma, will be released, which will provide the energy that will keep this reaction moving in the forward direction. And again, we are seeing another very nice picture here, which shows us the active structure, active site of the DNA polymerase. So I think we have a question. Okay, Gizem, type your question and then I will address it. So at the active site of the DNA polymerase, now if you are unable to see me, I would like you to open up your camera so you can see me. I'm showing here my left hand now, okay? So the DNA polymerase looks like the left hand palm of a human, okay? It resembles it. So when we look at this palm, there are some nucleotides there that are capable of binding to magnesium. And so remember, magnesium is absolutely essential for polymerization to happen. When we carry out PCRs, we always add magnesium to the PCR reaction. And this is why we add the magnesium, because the magnesium is positively charged and it can actually carry out an ionic interaction with the phosphates, with the alpha phosphate, and then also with the beta and the gamma phosphate, which will allow the 3' OH of the existing primer to come very, very close to the alpha phosphate so that it can attack this bond and generate that phosphodiester bond. So the magnesium that is found at the active site of the polymerase will actually bring the OH and the phosphate very close to each other so that the reaction can happen. Remember, I'll remind you at this point that catalysis will facilitate the reaction. The enzyme will facilitate the reaction, right? One other thing to keep in mind while you have your cameras open is when the polymerase carries out the actual polymerization, the phosphodiester bond formation, there is a major structural change in the polymerase whereby it closes in this manner. So it opens and it closes, and it opens and it closes. This opening and closing function of the polymerase also ensures another important characteristic of the polymerase, which is that it is very, very accurate. So when the polymerase closes like this, only the correct nucleotide, which in this picture is the adenine, which will base pair with the thymine, only when the adenine nucleotide has come will the polymerase be able to close very nicely, accommodate it within the active site, and then it will understand that the correct nucleotide has come, and therefore it will be able to catalyze the phosphodiester bond formation. If the incorrect nucleotide comes, a G comes, for example, it won't be able to close properly, and therefore that nucleotide will be kicked out. Now, in the event that an incorrect nucleotide does come in, these polymerases have a very interesting function, which is an exonuclease function. So they are actually capable of going back and cutting the phosphodiester bond so that the incorrect nucleotide is actually discarded. So even after the phosphodiester bond is formed, if there is an incorrect, if the polymerase senses that this is an incorrect nucleotide, it will activate an exonuclease function. This is a 3' to 5' exonuclease function, which will then go and it will cut the incorrect nucleotide, kick it out, and then allow the new nucleotide to come in. And this increases the accuracy of the polymerase tremendously. how did scientists detect the number of BPs that the polymerase synthesizes for each fragment? So this is detected with the help of radioactive nucleotides that are inserted. So when the radioactive nucleotides are inserted, then the signal of the radiation comes in certain lengths. And that is how it was identified that what the average length of those inserted sequences are. Okay. So DNA polymerase is working in this manner. We've already looked at this. We've already looked at leading strand, lagging strand. So here, and we've also learned about the primase, which will lay down the RNA primer, and we've also learned about the single strand DNA binding proteins. We've talked a little bit about the beta clamp, and we are already out of time. And what this beta clamp does is it loads the polymerase. So I'm actually going to have to stop here. We're out of time, unfortunately.